Our first faculty speaker is Dr. Sarah Dallas. Dr. Dallas is studying the dynamic interactions of bone cells here at UMKC, where she's director of the confocal microscopy core. She serves on the editorial board of the Journal of Bone and Mineral Research. But what you may not know about Sarah is she's, number one, she's from Britain, and she loves playing Celtic music on her Irish whistle. Now, she's not going to play today, but with St. Patrick's Day coming up, you might just imagine how she's going to unwind this afternoon after this is over. Sarah, come.
Right. Well, thank you for the opportunity to share some of our research with you. So for the past several years, the research focus of our lab has been to understand the cell-to-cell and cell matrix interactions of cells in the skeleton during growth, development, disease, and aging from a dynamic perspective. And the approach that we've used is to do live cell imaging or time-lapse microscopy in bone cell and organ culture models and in vivo to understand fundamental biology of the cells in the skeleton. So you might ask, why our focus on live cell imaging? Well, static imaging provides you a snapshot view of cells and tissues at a particular point in time, but live imaging enables us to visualize time-dependent changes in living specimens and can reveal novel biology and previously unknown mechanisms. Since many of you are not bone people, I think I should introduce you to the three major cell types in bone. First, we have the osteoclasts here, which are the multinucleated cells which resorb and remove bone, and they form from fusion of monocyte macrophage precursors. Then we have the osteoblast cells, which are these cuboidal cells on the surface of bone responsible for secreting bone matrix, which will then mineralize. And thirdly, we have the osteocytes, which are a subpopulation of osteoblasts that become embedded or entombed in this mineralized bone matrix. And you will notice that in bone, there's actually more extracellular matrix than there is actually cells. And this matrix is made from these banded collagen fibrils, which you can see here, type 1 collagen, which becomes mineralized with this black-looking hydroxyapatite on this electron micrograph. So I'm going to talk about our experimental approaches. For our imaging, we use a number of transgenic lines. And for the work I'm talking about today, we've used mice that have fluorescent tagged red osteoclasts using a TD tomato reporter driven by the LysM-Cree system. We also have mice that have a red fluorescent membrane in all of the cells. It's called the MTMG mouse. They have a red fluorescent membrane. And the third mouse is this GFP collagen tagged mouse here that we generated in my lab, where the collagen molecules are tagged with GFP. So these mice lines are crossed in various red, green, two-color combinations for live cell imaging of osteoclasts, osteoblasts, or collagen in various combinations. We do our live cell imaging with primary calvarial cell cultures or 8 to 14-day mouse calvarial bones from the skull, which are cultured in organ culture. And our time-lapse imaging is done either on a confocal or wide-field epifluorescent microscope. So I'm going to show you first some live imaging of collagen assembly by osteoblasts. But first, these are the mice we made that have this GFP-tagged collagen. So you can see it present in the dermis of the skin compared to a negative litamate. It's present in tendon, and nicely GFP collagen present in the bone tissue of this vertebra. Now, to image the collagen, we isolate primary osteoblasts from the calvaria of these mice. And if we grow them in culture on plastic dishes, we can get this nicely formed collagen matrix. If you give them ascorbic acid, if you don't give them ascorbic acid, the collagen stays inside the cell. So I'm going to show you a movie. One of the most informative models we've used has been this scoreline wounding assay, where we grow the cells in plastic dishes. We allow them to form this nicely organized collagen matrix. We then make an injury to the cell layer with a pipette tip to remove cells from this area, and we can watch them then migrating in and repairing the collagen. So the right-hand movie, you'll see the cells have their nuclei tagged with Hook stain. You can see the cells migrating in, filling in that wounded area, and starting to assemble collagen in this highly dynamic process in which you can see the wounded area repairing. Now, this next movie, I'm going to ask you to stop this when the cells on the right reach the bottom. So at this point, you can see the cells have migrated in. We're looking at one edge of the wound. They've completely filled that wounded area. They're confluent, but we haven't really seen much collagen assembly yet. If you could restart it or continue it. We see that once the cells become confluent is when we start to see collagen being assembled by these confluent cells here. You can see it being stretched and contracted by the cell motions. It's being pushed back as the cells advance forward. Very highly dynamic process with cells, waves of cells moving down, assembling collagen and adding to the collagen that was there before. Now, these movies are very data-rich, and we always feel it's important to try and quantitate some of these cell and matrix dynamics. And this is an example here where we took 10 early migrating cells that came out of the wound edge at the beginning of the movie, and 10 that migrated out about 48 hours later, and then tracked their motion trajectories. And this program also measures the GFP collagen fluorescence intensity under the tracking spot. So if we take all of those trajectories and start them at a zero origin in this 4-quadrant plot here, it's very clear that the motion is directional with everything moving towards the right-hand side of the plot. The tracks from the early migrating cells in blue are much more open and more directional, and we find that the late migrating cells have these more convoluted tracks where they even double back on themselves and they're not moving in such a directional manner. If we look at the velocities, they're dropping from about 30 microns to about 5 microns within this 90-hour period. And if we look at the speeds for individual cells that were tracked, it's very clear that both the average and maximum speeds are much lower in the late migrating cells. We're also finding an inverse relationship between the cell velocity and the collagen deposition, going along with the observation that they don't assemble collagen until they're confluent. So next, one of the things we observed was some interesting fibril dynamics from these movies. Here's an example here where a very actively collagen assembling cell here was split into 2 daughter cells, which then continue on down assembling collagen. And this was a novel observation because it was previously thought that the matrix-producing cells were post-mitotic. We've also observed what we consider to be fibril or a physical reorganization of the fibrils by cells. This is shown here, where, you can see that the fibrils start off as this much fine fibril meshwork, and then as the movie progresses, they condense down and get sort of physically squished and condensed to form this much thicker fibril bundle. And then on the right is very recent data from an individual cell with a red membrane, green collagen. You can see the collagen field around that cell that it's assembled, and it will split into 2 daughter cells, and you'll see them kind of rip apart the collagen matrix that cell had assembled as they move off in opposite directions. I think that one will play one more time. Hope you can see the cell sort of ripping apart its own collagen as the daughter cells come off. So for the last part of my talk, I wanted to move on to some recent work we've been doing of live imaging for osteoclast bone resorption and the real-time effects of drugs that are used to treat osteoporosis. So this is now a movie of osteoclasts, these big giant red cells like with this amoeboid motion moving on the bone surface. The shadowing that you can see on the right is actually the bone being removed, the collagen being removed by these osteoclasts. And we see a lot of interesting cell dynamics here, where a large osteoclast here splits into 2 daughter cells, and then there's a fusion event here where this osteoclast fuses with that other cell. So we see a lot of interesting cell dynamics, and we can look at the real-time dynamics of resorption. Now, inhibitors of rank ligand are used in patients to treat osteoporosis. It's clinically approved. The denosumab is an anti-rank ligand antibody, very effective for inhibiting bone resorption and increasing bone mineral density. But increasing numbers of patients have been experiencing rebound vertebral fractures due to a rapid increase in bone resorption when they come off the treatment. The denosumab doesn't work in mice, so we've had to use OPGFC as an alternative inhibitor of this protein rank ligand. So we wondered if our imaging of the real-time effects of these drugs could give us insight into what's happening in the patients. So our experimental setup here has a baseline imaging for 2 hours. I'm going to show you this treatment period. Thank you. I think it played a little sooner than I wanted, but I think you can see that even by just a couple of hours after treatment with this OPG, we're seeing this dramatic change in the shape of these osteoclasts. So the treatment period will be 44 hours. If you could restart the movie there. And you can see these cells have a very dramatic change in the morphology. We see little pieces breaking off and disintegrating, like here. And they're basically just sitting in place and not really moving in the same way that the actively resorbing cells are. And we also, you may not see these, but we don't see these kind of circular areas on the osteoclast representing the sealed zones where the resorption is occurring. We then removed the OPG and treat them with rank ligand to see if we can rescue the cells. I'm just going to go back so I can replay that movie. And I hope you'll see that they then start to fuse back together and they reestablish these little round structures, which are these actively resorbing sealed zones. And we can see that the resorption reinitiates. So we want to quantify this. We've looked at the morphology changes, and using thresholded and skeletonized images, we're able to show that there was an increase in the cell area, shown in blue here with the OPG, and the increase in the perimeter, a decrease in circularity, and an increase in the number of branches. And then lastly, this complicated slide is a way that we analyze all of these cellular dynamics, like the fissions and fusions. I have a brief time to explain. So the bottom line is the more complicated, these are fate maps of individual cells in control and treated bones. And the more complicated these diagrams look, it means there's more fissions and fusions going on. So we see about a 3-fold increase in the numbers of fissions and fusions of these treated cells. I didn't get a chance to show you, but the data from the collagen resorption dynamics show that the OPG completely wipes out resorption, but then we can restart it when we add the rank ligand at the end. So it's really just a flavor of some of the work we do. I'd like to say, in conclusion, I hope I've convinced you that live cell imaging can provide novel insight into the dynamic behavior of these bone cells, the mechanisms underlying bone formation and resorption, and responses of bone cells to pharmaceuticals. And I'll play my acknowledgements as movie credits while we have questions. Thank you.
Thank you. All right, time for you to participate with your questions. Are we getting any bites on the
Mentimeter?
QR code?
Yes.
And don't forget, there's a mic right here. This is too intimidating to be right out here in the front. Let's put it back here. That's not as intimidating. How's that? And we're going to let Dr. Arnold do the first question.
So I put it in Mentimeter, but I'm just going to ask it. So I saw you're interested in aging. We also do a lot of work with aging. So have you aged your models and looked at how these dynamics change with age? And if you have, at what age do you see that?
So we haven't, but we are doing work with age. It takes like 2 years to age up the mice to get them to what we would consider geriatric animals. So we haven't had a chance to do this imaging yet on the aged mice, but we are anticipating that some of those dynamics would be very different.
So one follow-up question. So we're interested in sarcopenia. And so sarcopenia and osteopenia and bone disease progress kind of together.
Right.
So if there's ever any interest in like looking how those are linked and collaborating, we should connect.
Yeah, so I've been a part of a program project with Linda Bonewald, a former faculty here, and it's all focused on looking at bone to muscle crosstalk and some of the soluble factors that mediate that. So
Great.
Yeah.
Thank you.
That was great, Sarah. I don't know enough about bone to ask intelligent questions, so I'll just make an observation. The NextGen team yesterday, we had the privilege of going to Linda Hall Library Rare Book Room, and so they showed us Robert Hooke's Micrographia, the first images of cells from
Must have been mind-blowing.
When was that? Late 1600s. So anyway, we've come a long way. It's pretty amazing.
Well, now we get instant gratification because we immediately see the images as soon as they're taken.
Again, I don't know enough about bone except for its histology.
Have you, has anybody else looked at the dynamics of bone and the response to decreasing estrogen as seen in postmenopausal women?
That's not something I've done yet, but the overoptimized mouse is a model that's quite commonly used in our field to look at bone loss, and there is a bone loss that occurs with that. So I would expect those osteoclasts to have increased activity.
Okay.
Okay, Mackenzie, let's get a question from,
Yeah, from Mentimeter. One of our questions is, what program or tool did you use to quantify the live imaging behavior?
Right, so this gives me a chance to talk about some new work that I didn't show today, but what I showed in the talk is, I would call it semi-automated. A lot of it's done in programs like ImageJ, where you have to click on the osteoclast each frame, and it's very time-consuming. I did have a slide on this, but didn't have time to cover it, but we're moving now to AI-based methods, which it's been a little tricky to get that to work in some of these movies because there's so much change over time, and some of the automated programs just don't handle change that well. So humans are still better in some regards, but now with the more sophisticated AI, we're able to, particularly with the movies that have the nuclei tagged, they can track the cells in a more automated fashion, and you can train them to recognize the same nucleus. And now we're able to get data from all the cells instead of just picking 10, and all the cells that are emerging from different times. And once you have that data, it gives you a lot more opportunities for looking at the interactions of cells and whether, when a cell meets another cell, if it alters its trajectory. And so there's a lot more data you can pull out of it.
all right, final question.
Oh, nice. Have you ever able to measure collagen on other structures that actually have them, like aorta or the vascular system?
So I'm a bit of a bonehead, so most of my research is on bone cells.
Was that a joke?
Yes. But the mice I showed you, I didn't get to show, but basically the GFP collagen is driven by a collagen 1 promoter.
Okay.
So it's expressed in all the tissues that normally express type 1 collagen. So we've got collaborators using it for fibrosis research. I think if you could use it to look at the collagen in the aorta, but we haven't done that.
Thank you.
Great, let's thank Dr. Dallas. And I love that we've already got our first collaboration possibility going here, so that's great.